Good afternoon. This is Kim McCleary. I'm President and CEO of the CFIDS Association of America, and I welcome everybody to our 5th webinar of our 2010 series. And thank everybody for joining us this afternoon. Just a few kind of points to get everybody oriented. I know for some people this is your first webinar, so we usually do a couple of housekeeping things at the beginning, and then I will introduce Dr. Suzanne Vernon, and she will lead us through a presentation about 3 of the association's 6 funded research projects, and also tying some of those investigators' approaches to studying CFS in with other research that's going on around the world. So again, welcome. I'm watching our attendee list kind of grow as the minutes pass 3 o'clock here on the East Coast, and I know we've got folks from literally most every time zone participating. So just a few things. Suzanne and I are actually in different locations. I'm in our office in Charlotte, and she is in her office in Colorado. So we can't see one another, and it's about as strange for us as it is for anybody who's sitting here looking at your computer and listening in. We're each alone with no body language to judge by, so bear with us as we try to orient ourselves using the various technology tools. We had just over 230 people register for today's program, and we have had tremendous participation in the other 4 programs that have preceded this one, not only on the day of the program, but also in terms of viewing of the recordings that are posted on our website and on our YouTube channel. And we are hopefully successfully recording this one as well, and we'll have it up later in the week for those who might want to view it again, or if you want to share it with somebody who wasn't able to take part during the live session. In our registration email, you were able to submit some questions in advance, and those have been shared with Dr. Vernon, and she will try to incorporate some of those in her presentation, but we will also have time for some questions submitted during the presentation at the end of her slideshow. So if everybody could just take a look at their screen, you should have a dashboard tool on the right-hand side of your screen that has a little dialog box to submit questions. Those actually will come into me, and then I will kind of sort through the ones that I can answer. Some of them are sort of simple about technology things and whether this will be recorded and that kind of thing, and others are of a more technical or specific nature to Suzanne's presentation. So I'll pass those on to her, and then we'll also have time at the end where I can ask some of the questions that come in during today's program. So if you have a question as she's going through, feel free to type it in so you don't forget it toward the end. And we'll try to get to as many as we can. We also get a log of all of the questions that are asked, and we'll try to answer even more of those through our upcoming publications, CFIDS link, and on our website and Facebook. And just as a reminder, while Suzanne is a doctor, she's a PhD doctor and not an MD doctor, and this format doesn't really enable us to address individual questions about symptoms, diagnosis, treatment, things like that, even when we do have an MD as our speaker, as we will later this month when Dr. Lapp joins us on the 20th. So if you could just hold off on questions of a more personal nature, because we won't be able to dispense any of that kind of medical advice. So without further ado, let me see if I can get my slides to advance, and hopefully we're not having any technical problems that I've seen so far, and move us along. Okay. Every once in a while we do have a little technical glitch on this end, and my slides don't seem to be advancing, so I don't have a picture of Suzanne to show, but I'm going to go ahead and introduce her anyway. Our speaker this afternoon is Dr. Suzanne Vernon, who is the CFIDS Association of America's scientific director. She holds her PhD in virology from the University of Wisconsin at Madison. And I'm sitting here still trying to advance my slides that aren't working. Sorry about that. But Suzanne joined our staff about 2 years ago when we expanded, dramatically expanded our research program. And we were just so fortunate to get somebody not only with Suzanne's background and training and 10 years of experience in CFS research, but also with her passion and ability to translate what is often a complex topic when you're talking about CFS research into language that is much more accessible to many of us who don't hold postgraduate degrees. And just a few more things about Suzanne to give you an idea of the breadth of activities that she undertakes in her role as scientific advisor. She's the co-author of nearly 80 studies in the published literature, including one that's going to be coming out fairly soon with one of the projects that you'll hear about today. She's a board member of the International Association for CFS/ME, the IACFS, that sponsors the biannual research conference that will be held next in Ottawa in 2011, I think. Suzanne is a member of the Department of Health and Human Services Blood XMRV Scientific Research Working Group. She's a member of the scientific committee for the first international XMRV workshop that was announced last week. She'll be representing the association next week at the International Society for Biological and Environmental Repositories meeting that will be held in Rotterdam. And she will also be giving a talk for the National Institute for Public Health and Environment in the Netherlands titled Determinants of Post-Infective Fatigue Syndrome Following Q Fever, which is a big problem in the Netherlands right now. And in addition to all of those roles, she is leading the launch and expansion of the Solve CFS Biobank that you'll hear a little bit more about. She's monitoring, collaborating with all 6 of our funded investigators, managing the research network that includes not only the principal investigators but all of their collaborators and a number of other investigators who have NIH support for their CFS research. And on top of that, she works with me on Capitol Hill to represent the need for expanded CFS research to lawmakers and public policy makers, and writes a lot of material for our newsletter, including a piece called Litmus Test that was in the link that went out yesterday. So we keep her fairly busy, and I think you'll join me in your appreciation for what she does by the end of this webinar. So Suzanne, I'm going to turn the controls over to you, and hopefully your slides will work better than mine did. I'll see if I can.
I have a little button that says show my screen, so I just clicked that. Can you see my screen now?
Not yet, but there is a bit of a delay. There we go. Okay.
Okay, now I'll try to click the slideshow button. There we go. Can you see that?
It's dark.
It should be a full screen that has my title slide on there.
It is dark at the moment, but I think there we go. Okay, CFIDS Association funded research projects is what we're all seeing.
Yes. Okay, super. And do I sound okay to you, Kim?
I can hear you.
Loud and clear?
Hopefully everybody else can. I haven't.
Welcome, everyone. These webinars are quite an experience. They're fun at the same time as being quite scary. It's not very often that I get to speak to large audiences, and these webinars give us the opportunity to speak to very large audiences spread out all over the country, and sometimes we even have participants from other parts of the world. So this is really fun, although every once in a while when I start thinking too much about all you people in various parts of the world, I get a little bit nervous. So I'm going to take a deep breath, sip of water, and take the next hour or so to tell you about some of our funded research projects. This is somewhat of an update. I'm going to tackle only 3 of our 6 funded research projects today. But I first want to start by telling you about our research program and really how it got started and how it was made possible. And in fact, our research program, which we launched as a new expanded initiative in 2008, $1 million research campaign, was made possible by donations from people with CFS, their families and supporters. I mean, that is absolutely amazing when you think about what you, the listeners, have done to make this research possible. Just a little reminder what we requested as far as research applications during this expanded research campaign that was launched in 2008. Now, a request for application is known as an RFA, and we advertised this RFA throughout, using all different types of media throughout the world, and we asked for research proposals that would advance the discovery of biomarkers and methods for early detection, objective diagnosis, and effective treatment of CFS. So as you can see, it was relatively broad. I mean, this is big and broad: early detection, objective diagnosis, and effective treatment, all things that are sorely needed in CFS. And what we got were 33 letters of intent, and after we reviewed those letters of intent, we invited 24 investigators to submit full proposals. Following that invitation, we actually received 21 full proposals that were acceptable and were able to be reviewed by a bunch of, actually 44, peer reviewers that were invited to participate in this review process. And these were experts in CFS science or in science in particular areas such as the autonomic nervous system or infectious disease or genomics. And these reviewers were also from all over the world. The top 9 proposals that were scored, that scored highest based on the scientific criteria, were then further evaluated for strategic merit. And let me go through some of the scientific criteria for that these proposals were reviewed on. Each proposal was reviewed and scored by 3 extramural or external reviewers, a primary reviewer, a secondary reviewer, and an ad hoc reviewer, in addition to myself. So again, each proposal was reviewed by 4 people. And I have 39 scientists here, and it's actually 44. Sorry about that typo. But there were these were people that were scientists, clinical researchers, and practitioners, all participated in this review process. And they were given various factors or criteria to evaluate the proposals on. And these included background and support for the hypothesis that they proposed, the objectives of the research, the study design and the methodology, data analysis and interpretation, importantly, a schedule and milestones, because these projects are limited to 18 months of funding. And whether or not they could do it, they could budget and justify that budget in that 18-month period. As far as what the qualification, in addition to what the qualifications of the investigator, institution, and research team were, whether or not they had approval to do human and or animal research on human and animal subjects, what the overall impact of the proposal would be, and finally the overall merit. These were all of the criteria that the reviewers scored the proposals on. So pretty rigorous criteria. again, once we had those top 9 scientific proposals or scientific merit proposals, they were then further evaluated based on strategic criteria. And these criteria are listed here. Whether or not the proposal actually satisfied the CFIDS Association program priorities. Again, those priorities were defined as objective diagnostics and improved treatments for CFS. And whether or not an end product or information useful to patients, the patients that made this research campaign possible. That was also another important criteria that we evaluated. In addition, whether or not the research project filled gaps in the existing knowledge for CFS based on objective diagnostics and treatments. Whether or not the proposal, the research project, had the possibility of securing greater funding. After we funded it, would it produce material results that would be attractive to other funding agencies such as the NIH? And finally, whether or not the project would resonate with our supporters. And the result was we funded, of those 9 top-scoring proposals, 6 projects were funded. And these included Gordon Broderick up in Canada at the University of Alberta, Sanjay Shukla at Marshfield Medical Research Foundation in Marshfield, Wisconsin, Bud Mishra at NYU, New York University in New York, Marvin Meadow at New York Medical College in New York, Jacoma Shangu at Weill Cornell Medical College in New York also, and Kathy Light at the University of Utah Health Sciences Center in Salt Lake City, Utah. Today I'm going to talk about these 3 investigators, or these three research projects that are circled: Gordon Broderick, Sanjay Shukla, and Bud Mishra. I'm going to start off talking a little bit about Sanjay Shukla's project, which deals with the microbiome. Now, I don't know how many of you have heard of the microbiome, but it is now considered our other genome. Really pictured the cover picture of Nature magazine, one of the top science magazines, that featured the microbiome, or our other genome, on the cover of Nature in just, I think it was the March 4th issue, just this year. The microbiome is basically the genomes, the combined genomes of all the microbes that live inside and on our bodies. We now know that there are 10 times more microbes than human cells, and that translates to greater than 3 million genes in the gut. Actually, that's only the gut, the colored part of this picture to the left. 3 million genes. That is 150 times the human gene complement. So this is quite a genetic contribution to our own genome, as you can imagine. This in its entirety, the microbial genes, the microbes and their contributing genetic products, as well as our genome, is known as a metagenome. What Sanjay will be researching, or is currently researching, is the microbiome. All of the microbial genes in the gut in people with CFS. Let's find out why. Well, actually, let's talk a little bit about who would have the nerve, or maybe the audacity, I guess, to tackle such a tremendous problem, complex problem. To the right, up here at the top right, is pictured Sanjay Shukla. He's a PhD microbiologist at the Marshfield Clinic Research Foundation. He's the PI on this project. His team that he has recruited includes Steve Yale, who is an internist also at Marshfield. Dane Cook, who some of you may be familiar with. Dane worked in New Jersey with Ben Nadelson and Gudrun Lang for a number of years prior to joining the faculty at the University of Wisconsin in Madison. And he has also recruited Dan Frank, who's at the University of Colorado Boulder. Sanjay, again, the PI, is the microbiologist with the microbial expertise. Steve will actually be doing most of the clinical perspective of this project. Dane is an exercise physiologist by training, and he will be actually testing all of the patients and controls in this study. And Dan Frank is the one who is going to be cataloging all of the microbial genes that are identified, collected in this study.
Hey, Suzanne, I would just also add that Dane Cook has recently been appointed to the federal CFS advisory committee that will be meeting next week. It'll be his first meeting, but we had nominated Dane to the committee, and we're delighted that he's on it now and brings his exercise physiology and interest in post-exertional relapse to the committee's deliberations.
That's a great point. And Dane is also one of the best study design experts I have encountered during my professional career. He has incredible attention to study detail, and this study is no exception. So this is going to be a very exciting study. And in case I forget, he also was just awarded about $30,000 from the University of Wisconsin as an add-on, as an adjunct, as a supplement to this particular study. This is going to allow them to enroll more patients and also to hire, I think it's 2 research assistants to participate in this study. So quite an impressive undertaking going on here. The microbiome is huge. It is so huge. Well, again, as you can imagine, 3 million genes, 150 times the human genome complement, and yet we know very little about this huge microbial mass that we have living on and in us. The difference between microbes or bacteria and viruses is that you can see most of the bacteria underneath the microscope, and that's pictured here in the microscope, and then on to the right, all the different types or shapes of bacteria that we know exist. The conventional way of looking at bacteria is to culture them, and that's culturing them on these jello-like or agar plates. This is challenging because it's very costly. Many of the bacteria actually don't grow, or we don't know the types of media that they can grow on. And sometimes the bacteria take very, very long periods of time to actually grow. And in reality, the majority of the bacteria cannot be cultivated for reasons that we don't know what their temperature is, what kind of nutrients they require, etc. So the new approach for cataloging this microbial biomass is by using DNA-based techniques. We harvest the sample that we suspect contains the DNA from the bacteria, isolate the DNA, and then sequence this. And this is what has been done and what will be done in this study in order to catalog all of the bacteria in the gut. What influences the human microbiome? Now, I think it's important that we realize that these microbes, the microbiome, is there for a reason. It is thought of as a core, an essential component to our health and well-being. We modify our human microbiome, or our gut microbiome in particular, by our lifestyle, that is the things that we eat, the things that we do, the medicines that we take, etc., by other diseases that we might also have throughout our lifetime, by the types of environments that we live in, by our own genotype. Remember, we have a human genome complement that's interacting with this microbial complement all the time, by our ongoing physiologic processes, whether or not we, the certain types of work that we do, the other things that affect our physiology, such as diet and exercise, and again, our immune system will also affect the microbiome. If we get sick with the flu, that has an influence on the types of things that go on with the microbiome in our gut. In addition, we also encounter various transient types of microbial encounters that will affect our gut microbiome. So as you can see, there are a number of types of things that can affect this core microbiome that is essential for balance, health, and well-being. It has been suspected for years now that there are infectious agents that are either the cause or can trigger CFS. We know that some of the symptoms of CFS, such as fatigue, diarrhea, low-grade inflammation, pain, and fatigue, could potentially be explained by alterations in what is going on in our gut. And we know that people with CFS have high rates of gastrointestinal disturbances, or what has also been called leaky gut syndrome. So it is possible that an altered microbiome in people with CFS could explain some, or help explain at least, some of these symptoms that are common in people with CFS. We already know that altered microbiomes explain some other complex diseases, including ulcerative colitis, bacterial vaginosis, and obesity. Now, it's important to also understand that these characterized diseases, where the microbiome has actually been characterized, do not, I guess, the microbiome does not work by itself. In fact, there are human genome or human genetic components that interact with the microbiome to result in various aspects of these diseases. And it's suspected that that could be the same thing in CFS. So what Sanjay and his team are asking is, could CFS be a disorder in which infectious agents may affect a change in the immune system, causing the CFS symptoms to appear and to persist? They're going to examine this using an exercise challenge paradigm, because we know that post-exertion fatigue is unique. It's in fact a unique feature to CFS, and it is a distinguishing factor from CFS, distinguishing it from other illnesses, including depression, rheumatoid arthritis, multiple sclerosis, and cardiovascular diseases. So any research that's done in CFS, and this is a paradigm that they've chosen to include in their study design, needs to be put into a post-exertion context. Let me make sure I didn't jump ahead here. There we go. So hopefully you can see now the slide that shows, it was entitled The Microbiome and CFS. And I put this slide up here rather than a more complicated molecular slide to give you an idea of what their study will do. This is a nice magnetic image of the abdomen showing the gut outlined here. What their study will examine is individuals that have CFS and perhaps have a genetic predisposition. They're going to be screening for certain factors, that these individuals with CFS have an altered microbial ecology in their gut. That is, they have either a different ratio of good bacteria to bad bacteria, and that altered ratio will increase the permeability of the gut, allowing the bacteria that are normally supposed to be inside the intestinal wall to escape out or translocate into the bloodstream. And that with exercise, this translocation of the bacteria across the gut is actually increased. This then triggers a low-grade inflammation because of the increased level of bacterial products called endotoxins in the blood, which is a signal to the body or to the immune system to respond. Their specific aims are to determine whether or not CFS patients harbor an altered community of microbes compared to healthy control subjects. And you'll see this on this study diagram to the right, where they compare a CFS patient matched on certain immune genes and sex and age and race to a control subject. And also, because this is an exercise challenge study, these subjects will be matched on condition. They will take a blood supply or a blood sample and a stool sample before the subjects are exercised, and again subsequently at 1 hour, 48 hours, and 72 hours afterwards. This is blood. A stool sample will be taken at 0 hours and 72 hours after exercise. And this will happen in both CFS cases, patients, and controls. All of these samples will then be analyzed for the bacterial products that are present in the samples. In addition, the blood samples will be analyzed for cytokines. All of this information will then be analyzed by sequence and for the various cytokines in order to get an idea of the inflammatory load in the CFS patients compared to the controls. This is, again, all done using standardized criteria, a standardized exercise challenge, and this study is currently enrolling research subjects. It's happening in Wisconsin, so if you are in Wisconsin and are interested in participating in this study, you can contact us and we can put you in contact with the investigators. This, because of the complexity of this particular study, it is now, it has actually only gotten approval and underway. I think they started enrolling people in February. So we hope to have the first sets of results in the coming months. And I think this is just an exciting opportunity and the first study of its kind in CFS. So state-of-the-art, first of its kind, very exciting, and I think in many ways will provide us with a lot of information about what is going on with CFS. And as you can imagine, if it turns out that there is an altered microbiome in people with CFS, there are ways that are available to us to be able to modify that or fix that. So this is a project that's not only giving us information about pathophysiology, but also giving us possibilities for treatment of people with CFS. The next study I would like to update you on is the study by Gordon Broderick at the University of Alberta. This project is titled Molecular Patterns of Persistent Immune Activation in a Post-Infectious Adolescent Cohort. Again, this is a team of investigators, Gordon Broderick being the principal investigator. He is in the Department of Medicine at the University of Alberta and is actually an electrical engineer by training who has turned his skills in computation to actually computational biology or a systems biology expertise. Ben Katz is his collaborator, and Ben is a pediatrician with expertise in infectious disease at Northwestern University. Renee Taylor is a psychologist at the University of Illinois in Chicago. Carl Schaefer is also a computational biologist at the NCICB, which is the Computational Institute at NIH. And Saul Afrani is at the Bar Ilan University in Israel, and Saul is also a computational biologist. So again, a very interesting and powerful team of investigators with a variety of expertise. I think that's really needed to tackle a CFS or post-infectious CFS from a systems perspective. So this study is looking at what happens, again, predictors of someone developing CFS following infection. That's this little picture here with all the nasty-looking microbes on the person that's sick in bed. It's focusing on one particular type of pathogen, and that's Epstein-Barr virus. And this is an electron micrograph of the actual virus particles that are characteristic of Epstein-Barr virus. And then this lower picture is a picture of Epstein-Barr virus in the blood. So this is a study of post-infection Epstein-Barr virus in the blood. What is going on? Can we identify predictors, and can we detect it early? In other words, can we stop CFS before it becomes CFS? That's what this project is about. Gordon has a systems biology perspective of human systems. And I guess this probably comes from his systems engineering background. He believes, and I think this is very true, most biologists, I believe, think this way, that the human body functions as a highly integrated system. They're distinct systems, but they're still tightly integrated. In other words, your brain and your immune system and your endocrine system are distinct systems, but they all function together in harmony, integrated as illustrated by these cogs in a wheel. And when there is harmony and balance in this integrated system, you have stable and efficient energy utilization. However, when there are of insults to the system, whether it be to the central nervous system, such as could happen with head trauma, or various types of emotional stressors such as loss or death or various types of injuries, or whether there be other types of stressors such as an immune stress, which occurs with infection or vaccination. These are insults or assaults to a highly integrated system. What happens is the integration comes out of balance, and there's no longer an efficient use of energy. It's unstable, and it's inefficient. These are the things that Gordon is attempting to measure. why would we measure these types of things in a post-infectious EBV cohort? Well, first of all, this particular project is leveraging an existing study. And this study was actually one that was funded by NIH. Dr. Renee Taylor was awarded an NIH grant or award to study post-infection EBV. And Gordon was aware of this project award and assembled a team or proposed this particular project with Renee and colleagues in order to tap into and further analyze the post-infectious cohort. EBV is known to cause or trigger CFS, and therefore it serves as an excellent model system to study the triggers or the potential causes or the predictors of what is going to cause or determine those who get CFS. Infectious onset, such as one that occurs with infectious mononucleosis following EBV infection, is a very easily characterized event or insult to a system. We know the kids that get, or the people that get EBV-associated infectious mono because of some of the measurements that we can take. This study also was a very large patient cohort that had extensive clinical characterization and other various types of measures taken. And they were assessed over a two-year period. So as you can see, it's an incredibly rich data set, and it was a funded study, so we're really leveraging a very large resource in order to further understand what's going on in this post-infectious cohort. Let me tell you a little bit about the cohort itself. Originally, 301 adolescents got sick with infectious mononucleosis, and it was confirmed to be caused by Epstein-Barr virus. Of that 321, where's my mouse? 231 of those adolescents recovered. 70 of these people were recruited into the next phase. So this is considered baseline. Again, these were acute EBV-confirmed infectious mononucleosis subjects. 70 of those were then followed at 6 months, and at that 6-month period, 39 of those were confirmed with CFS. That's 13% of the initial amount. Of that, 31 recovered. If we move on to 12 months, 53 of those were then recruited or enrolled or followed into the 12-month period. By 1 year, 22, or 7% of the original infectious cohort had CFS, and 31 had recovered. And finally, by 24 months, 13 of the original had CFS, and 40 had recovered. Now, all of these subjects met CFS criteria according to the 1994 case definition, and since they were adolescents, they also met the pediatric case definition that was developed by Lenny Jason at DePaul. Over time, these subjects had blood collected, which was used for Gordon's study for gene expression analysis and also cytokine analysis. And all of this information was combined with the clinical and other experimental data that was collected over this 2-year period. So what can Gordon do with this type of information? Gordon has developed computational techniques, and this is what I'm showing under the study methods slide, that is capable of combining clinical information that is collected from these post-infectious fatigue subjects, as well as the type of clinical information that is collected in many or all of the CFS studies that we conduct, combines that with the various types of biologic measures, and can then use this information to differentiate people with CFS from people that actually have insufficient symptoms or fatigue, so they're not defined as CFS, from those people that are not fatigued, or the healthy controls. So this is something that is a very powerful, potentially diagnostic method that uses both clinical information as well as biologic measures in order to be able to classify or differentiate people with CFS from those that do not have CFS. Let's dig a little bit deeper and see exactly what happens, how he can do that. Now remember, again, I told you that it's not just one thing that Gordon or anyone can measure in order to be able to differentiate people with CFS from those without. It's actually many different types of variables. And what Gordon has found using these approaches that he has developed is that immune information together with other types of physiologic measures are altered in people with CFS. So on the right, you'll see this spider web of signals. These are actually endocrine measures and immune measures and other types of physiologic measures from people with CFS. This is not the post-infectious fatigue cohort yet. This is what he applied his techniques to people that have known CFS. This is actually from the Wichita cohort from the CDC study. And you can see over here that this network or this spider web is different from people that do not have CFS. If you just focus in on this highlighted blue box, we see a number of immune cells that are actually kind of talking to one another over here, and a few hormones or endocrine molecules, such as NPY and ACTH, all talking in a specific fashion in the non-fatigued individuals, in contrast to the people with CFS, where we have a different communication network or a different signaling pattern. Some of the same players there, but now they're not talking to the same cells or molecules that the non-fatigue cohort is. So these are the types of computational approaches. This is called an information network analysis, taking measures and information from each one of these measures, combining it in a very sophisticated way in order to be able to identify the differences in what's going on at a very molecular, low-level way between CFS and those that do not have CFS. This is the technique that he's going to apply to the same samples in the post-infectious cohort, which the data from that, from the blood gene expression and the cytokine analysis, is currently being generated at Northwestern University. But in the meantime, Gordon has not been idle. He just heard today that a paper applying this technology to CFS was accepted in a very impressive journal called Brain Behavior and Immunity. This paper is in press and will be available next week, I believe. And as you can see at the bottom, that's Gordon as the first author, and he partnered with Jim and Andrea, myself, and Nancy and Marianne, Nancy Klimas and Marianne Fletcher at University of Alberta. The cool thing about this particular application of this network mutual information technology to CFS on this particular cohort, which was from Nancy's clinic, showed that the observations are consistent with several processes active in latent viral infection and would not have been uncovered by assessing one marker expression alone. So, again, to emphasize, it's really a combination of bits of information or mutual information that will get us to really understanding and targeting some of the differences that are occurring between people with CFS and those that do not have it. So the anticipated outcomes from Gordon's study are as follows. We anticipate getting closer to objective diagnosis by isolating patterns or these motifs that will get us to a robust diagnostic pattern indicative of CFS. This graph here shows a timeline, our baseline samples for post-infective fatigue, 6 months, 12 months, 24 months. And these little networks down below, each one of these, indicates the types of information that he will be collecting at that has been collected at each one of these time points, and then analyzing them. So you can imagine that at 24 months, this could be a pattern that is objective and, in fact, diagnostic of someone that has CFS or has the potential to recover. And this is put in the context of various types of symptoms that the individual experiences over time. It also has the potential to detect CFS early by looking at these baseline patterns. By looking at baseline, we could potentially be predictive of who is going to recover or who is at risk of developing CFS over time. Remember that the earlier CFS can be diagnosed, not waiting until 6 months, not waiting until 12 months, but an objective early diagnosis gives a greater opportunity for, number one, not even developing CFS or dealing with it, intervening much earlier with a greater chance of recovery. So again, some exciting anticipated outcomes from a very exciting approach that is being applied to CFS, or a model system of CFS, and that's the post-infective cohort. But you can imagine that if we can develop these types of tests on a blood sample, which is easily obtained, that something like this could be adapted to clinical practice. Finally, I want to wrap up by talking a little bit about the work that's being done at New York University. But I want to start by emphasizing some of the challenges that we face, not only in CFS, but in medical research in general. Pictured on the left here are a bunch of different scientists, investigators, sitting at their computers, plugging in all the results on the research that they've done. This has actually happened in CFS over the past 20-plus years, and I think it has allowed us to develop a pretty comprehensive picture of what is going on in CFS. We know some of the risk factors for CFS. We know the various types of systems. We know the players that interact in order to affect the HPA axis, to affect the cardiovascular system, to affect the immune system, the gastrointestinal tract, the musculoskeletal tract. We have a pretty good, excuse me, a pretty good diagram of what's going on as far as the pathophysiology of CFS. Yet, we do not have a good sense of where we should aim. Where do we target in order to be able to get that objective diagnostic test that we need for CFS? Where do we target in order to be able to treat individuals with CFS? We don't know that. How can we possibly take all of this information that has been gathered in order to help us find the appropriate target or targets? This is a challenge. Well, someone who's actually, again, rising to this challenge is Dr. Bud Mishra. Bud Mishra is at New York University School of Medicine. He is a mathematician, computer scientist, philosopher, and one that is not afraid to try to use computational science in order to be able to describe biological issues. So he's applying his expertise to CFS. He's collaborating with Eric Aslaksen, who was a colleague of mine actually at the Centers for Disease Control and Prevention. Eric is a physicist and now working with Bud in order to be able to put their computational skills to the test as follows. This is what we've called kind of the Google for CFS approach to identifying possible causes, as well as consolidating information in order to be able to understand CFS. And again, when I say understand, I mean to get at objective diagnosis and to be able to target treatments. This is the way that they are doing this. Follow my pointer up here, and to the upper left you see a pile of papers that's called Cell, and another pile of papers called PNAS. These are actual journals. So what Eric and Bud have done is to create a gigantic library by downloading or by creating this library from all of the medical and science journals available to us in PubMed. PubMed is the National Library of Medicine's medical and science repository. So they have grabbed essentially all of these information, both in abstract form and in full text, and created a library from it, their own library. What they then do is they convert this into a form that can then be searched using various types of text mining tools. There's some approaches that they use that can actually read articles. These are natural language processing tools that can read and find words and then extract patterns or various types of indicators that you tell it to in order to be able to find these patterns that are of interest. So for example, for us, we're interested in CFS. But we're not just interested in only CFS articles or information. We're interested in, let's say, for example, a genetic marker that has been somewhere in the medical literature associated with, I won't say fatigue, let's say cardiovascular disorder or autonomic nervous system disorder. One of the indicators of CFS that has been consistently found, heart rate variability, for example. Anyway, all of this information is gathered into one place, and mined in such a way to pull out patterns that can then inform us about what is going on in CFS. Not only what is going on, describing that picture of pathophysiology that you saw in the previous slide, but also give us indications of what actually might be causing that problem. That is the goal of this. Where these investigators are at right now is at this phase. They have created a gigantic library that is now extracting patterns that is explaining CFS. They combine that then with the experimental data that we have also gathered on CFS. And these are experimental data points from our funded investigators, as well as experimental data points from other data sets that are publicly available. For example, the Microbiome catalog is publicly available. And this information, combined with the other published literature and the information that we get on CFS, is then used to identify various markers that can be used for diagnosis and or treatment, and perhaps identify possible causes of these. And this is illustrated in this next diagram. This is an actual simulated data set that Eric put together, and what it shows is an output of this procedure. So all of these red dots here are papers or are results or information that are associated with the gut. All of these data points, and this could be a genetic marker, a physiologic marker, a bacterial marker. All of these are associated with the gut. The same here for the brain. They're all tightly networked, tightly correlated, so that these are all indicators of the brain. Down here, all indicators of the blood. Each one of these dots representing some bit of information specific to the blood. And this is a hypothetical diagram, but from actual simulated data. And here, this one point that ties everything together would be, for example, a genetic marker, or could be, for example, a specific virus. But this is what this project has the potential to give us. And again, this is a very exciting research project that is essentially the equivalent of our Google for CFS project. So I think from this update, hopefully you are convinced or persuaded to believe that together we can solve CFS. And I'd like to show this diagram in most of my presentations because I believe this is a very achievable paradigm in order to get from screening very heterogeneous populations into more homogeneous groups, including isolating out people with CFS based on objective markers, such as some of the ones that I've told you about or that we're poised to discover. Further dividing this CFS pie into more discrete molecular subtypes, which can then be very closely targeted in order to make interventions, pharmacogenomic interventions or pharmacologic interventions, all the more effective and targeted. This is something that we can achieve together with, I'm sure of it. We can solve CFS, folks, and again, all of the research that I've talked to you about today and in the past has been made possible by you. Many of you have asked questions, and those of you that registered asked whether or not you could participate in research. There was one project today that I told you that if you live near Wisconsin and you were interested in participating in that particular project, you have that opportunity. We also now have the Solve CFS Biobank, which is going to give many people with CFS the opportunity to participate in research, to be an invaluable resource in order to really advance the research and get us closer to these objective indicators that we so badly need in order to be able to continue to advance our research progress. Remember, we did this. We started out just 2 years ago with a $1 million campaign, and I hope you can appreciate the value that this research really does show have for that kind of money. We have 6 investigators, 6 projects that we funded, but really what we're getting are about 25 investigators funded, and we're painting a picture. We're gathering data from many parts of the body, and I think these parts brought together, which is what we can do by continuing to support this type of research, are really going to give us the best indication of what is going on with people with CFS in order to really get us to an objective diagnosis and targeted treatment. So I hope you will continue your support of the CFIDS Association, continue your support of us in order to support this, these impressive investigators and the really important research that is being done. Go to our website, www.solvecfs.org or www.cfids.org, and there is a lot of information, a lot of resources present on both of these pages that can that you can have, that you can get, and that you can also push that nice little green button that says donate now to help us raise the money that we need in order to continue our support of this research program. I want to thank you for your time and attention, and I think I'll turn over the audio now to Kim.
Okay. Are you going to also shoot me the controls? Maybe I can do that.
If I can do that.
There, I think I've...
You are no longer the presenter.
Okay. Hopefully. There we go. Just want to echo what Suzanne said, that none of this research would be possible without the support of individual donors who made it all entirely possible, and all of the people that she described as being involved on the front end of making the very challenging decisions about which studies we would support through our research program. And I know I've gotten a couple of comments from people who are just so impressed, Suzanne, with your ability to sort of bring all of this very complicated information down to a level that most of us can understand. And I appreciate the audience hanging with us. We've almost gone into the full hour and 15 minutes that we planned, but we do have some questions that I'd like you to answer if you could. The first one is, and I tried to answer this through the little chat box, but probably other people have the same question, as to what you mean specifically by the term objective diagnosis. How does that differ from the situation that we have currently, where the diagnosis of CFS is made based on symptoms and the exclusion of other conditions that might also be responsible for symptoms that are quite common?
Okay, so that's key right there. The symptoms that characterize CFS are in fact quite common. They're nonspecific, and they're reported by the individuals that suffer from them. This, of course, is subjective. It's something that you describe to a practitioner. If we had an objective measure or an objective indicator, something that we could measure, whether it be in a stool sample, for example, or in a blood sample, or in a saliva sample, that is what I mean by objective diagnosis. It's a blood test. It's like what a sugar test, for example, like they have with diabetes, where you measure the amount of sugar that's in your blood or not. So that is an objective test. With that, we can do away with all of the subjective indicators of these common symptom descriptions in order to be able to really target the types of interventions that we need. Does that make sense? Kim?
I think so. I think that's sort of, I don't know, prognosticating to your part 2 of this look at our 6 funded programs, imaging techniques and exercise models may also yield diagnostic tests. So it's not just limited to blood, urine, stool samples, things that are testable under lab or microbiology type examples, but also things like the MRI that's used to help diagnose MS and different cardiovascular challenges that might show up in cardiovascular disease. So
exactly, yep.
Looking at all those things as well. Another question
that we can measure,
right,
is basically it.
Without any sort of opinion needing to factor into that for the non-believers that are out there. Another question.
I'd like to add too that. Let me add one more thing. I guess it was about 1.5 years ago, or maybe it was from our Medscape module. We asked what would it take for practitioners to be able to diagnose and deal with CFS, and the number one answer was an objective measure, something that they could basically use to objectively evaluate the patient. So it's a very important measure. You know, in our current healthcare settings, where we have so little time in order to be able to, have face time with a doctor, something objective that really zooms in on what the problem is sorely needed for CFS.
And the other piece of an objective measure is also measuring the effect of treatments. Right now, the only thing we have to show that treatments work is the patient's evaluation and the physician's evaluation of how much better they feel, which is obviously important, but pharma companies like to also be able to measure that somehow. And so that'll be a huge advance forward. Suzanne, another question that came in, I think actually a couple of questions that came in relate to the study of CFS cases and CFS and controls that are either healthy or have other conditions, and why, will you explain why it's important to study CFS not just in isolation but in contrast to the controls, whether it's the EBV study where you're looking at people who had acute Epstein-Barr virus infection and mono and recovered from that infection versus the folks who didn't, or whether it's the Dr. Shukla's study where he's looking at the microbiome in CFS cases versus matched controls.
So the studies, those two studies are case control study designs. So in that case, it's important to compare someone who stayed sick following Epstein-Barr virus infection and infectious mononucleosis compared to someone who recovered from that. And it's kind of like subtracting out the differences or finding the differences. The only way that you're going to be able to determine that something is a cause or an effect is by comparing to something that doesn't have that. So that's why the other study, for example, compares the gut microbiome to a healthy control. Everything is kept the same, except, or as similar as possible, except the disease state. So in the case of Sanjay Shukla's study, the controls will be neighborhood controls, so somebody that lives in the nearby vicinity, and not necessarily comparing somebody who lives in Little Italy to Chinatown, for example, in which case you might suspect that those differences would certainly influence what microbes are in your gut. So healthy controls in the same neighborhood or the same vicinity, same age, same sex, so that the differences that we find in the gut bacteria are most likely due to the illness or causing the illness and not by something else. That's why you have a control, so that you can kind of subtract out all of the differences. There are other ways to other study designs also where you can study an individual over time, one individual, and in that case, time is the necessary factor, and you can assess or compare what changes in a certain individual over time. As you can imagine, that is a tough study to do because it's hard to follow individuals over time, even though there are both birth cohorts, but certainly possible. It just so happens that these two studies that I talked about today are case control study designs.
Very good. Question that came in was, are you also searching for the effects or the role of heavy metals in infection and dysfunctions in the gut in Dr. Shukla's study? Do you know if their analysis will be able to make some sense of that issue?
They are doing the required blood workup. I do not believe they are looking at any of the various types of metabolites or products in the urine, and I do not believe that they are targeting any of the heavy metals. But they are doing the required blood workup, and yeah, so I would say no, they're not. They are not looking specifically at heavy metals. They are just cataloging the microbiome in CFS patients following exercise challenge compared to healthy controls, and also looking at cytokines.
Well, and what's so impressive to me is that that whole area is certainly not going to be settled or understood with one study, but it's a fascinating avenue that's never been explored before in CFS, but fits in with so many of the symptoms that are very characteristic.
That's right. I mean, and the thing that's very exciting about this study to me is that we know that there are energy differences in CFS, metabolic differences in CFS, and the microbiome is an essential component to having balanced energy and metabolic processes go on in our body. So even if we can determine whether there's a difference between CFS and healthy controls. I mean, that's a huge step forward, a huge step forward.
I can hardly... Oh, I know. There was another question about Epstein-Barr virus, and the question related to the fact that very early on EBV was sort of ruled out or dismissed as a potential cause of CFS. So why does EBV continue to be a topic of interest in CFS research?
EBV is a common viral agent that 95% of the human population is infected with by the time we're in our mid-20s, early 30s. So almost everybody has EBV. I think the question is better phrased as, why do only certain people that get infected with EBV and get infectious mononucleosis not recover, and in fact develop the equivalent of CFS? And I think we know now, or we can appreciate now, that there are certain individuals in these that develop this post-infectious EBV CFS that have certain vulnerability traits, and that would be genetic vulnerability traits that alter the way our immune system may handle the virus that in fact all of us have to live with for the rest of our lives. Not only are there some host genome vulnerability factors, but the virus in fact has evolved in such a way to essentially hijack some aspects of our immune system. It encodes proteins that mimic some of our own immune processes, such as complement activation, for example. It encodes receptors that affect the way our immune cells function. Certain ion channel receptors can be encoded or manipulated by the virus. So I think it's not just a cause and effect thing. It's more of a lack of synergy between a virus that infects all of us and certain people because of various aspects that make some individuals more vulnerable than others. And those are the types of things that studies like Gordon's post-infectious fatigue study can help us identify. Why do some people not recover following EBV and develop CFS compared to others?
Yeah, I think if you talk to researchers like Ron Glaser, University or at Ohio State, who have studied EBV for most of their careers, EBV was sort of underappreciated 20 years ago, and now with newer tools and more sophisticated ways to look at this virus, I think we'll continue to hear a lot more about it, not only in CFS but in other disease states as well.
I'm really glad you brought that up, because Dr. Glaser just had a very important paper published in this week's issue of Brain Behavior and Immunity that shows how how certain molecules of our endocrine system interact with Epstein-Barr virus to allow the Epstein-Barr virus, which is normally quiet or latent in most of us, to become activated. So here is a perfect example of the interaction between a virus which isn't supposed to be doing anything in our body, just kind of laying quiet, and our body and how it responds to certain types of signals from the endocrine and immune system.
I think that's a great place to end, and I can hardly believe I'm going to say this, but we got through most of the almost all of the questions. We've gone a bit over our time, and I just want to put in a plug for a couple things here. Our continuing webinar series, and we're kind of having a double header this week with today's program and another one tomorrow on conditions that frequently coexist or overlap with CFS, and we've got speakers who are very knowledgeable about some other conditions that many people, particularly women with CFS, deal with in addition to their CFS. And that program is tomorrow at 3 p.m. Eastern Time. And then on May 20th, we will have Dr. Chuck Lapp from the Hunter Hopkins Center in Charlotte sharing with us his approach to treating CFS and fibromyalgia. And many of you know Dr. Lapp from his many years of caring for CFS patients and being on the IACFS-ME and the CFS Advisory Committee and a number of other high-profile positions to which he's made a lot of important contributions. And then on June 8th, Suzanne will be back with us with Liz Horn of the Genetic Alliance to talk about the Solve CFS Biobank. And Suzanne will wrap up with the other 3 studies among the 6 that we fund later in June, and I foolishly did not bring that date with me, but I believe it is the 3rd Thursday in June, but you can check our website for that. And yeah, there we go. And I just also, Suzanne had mentioned the biobank, and just wanted to quickly give you some information about the Solve CFS Biobank. If you have not already read our announcement and some of the materials on the website that Suzanne referred you to, we'll include those in the follow-up message, some URLs for you to check out. But it's a very exciting project that Suzanne, together with the folks at the Genetic Alliance Biobank, have developed that will make it possible for a wide variety of people to participate directly in the research and in compliance with many important standards for patient privacy and data security that are offered through our partnership with Genetic Alliance. We are recruiting for the first study right now, and that for just the inaugural study is limited to 4 physicians to ensure that we have firm clinical diagnoses and well-characterized samples, as Suzanne has already discussed earlier, how important that is to be able to facilitate research. But we will hopefully, with the expansion of funding, be able to expand participation to a wider group of patients than currently meet the enrollment criteria. And this is just an incredibly important tool to advance research so that we have this biobank of samples and clinical information to facilitate all of the types of research studies you heard about today, in addition to research that's going on in other labs and academic centers around the country. So with that, I think we will end here and just appreciate the fact that so many of you hung in with us for the full now 90 minutes, and hope you'll be able to join us tomorrow or on May 20th or for one of the other upcoming webinars. Appreciate the great questions that came in, and hope you'll take just a minute after the webinar ends to fill out a short survey that your screen will automatically pop to. So thank you, Suzanne, for another brilliant presentation and for bringing it down to our level, all this great research and science that's happening. Thanks, everybody, and we will see you back tomorrow afternoon, I hope. Take care.
Bye.
Bye.